Hello there, fellas. I am Alanna Nicole, and for today's discussion, we will be talking about embryo transfer. What is embryo transfer? Embryo transfer is an important technique as it provides avenues for reproductive and genetic enhancement. It is a phase in the process of assisted reproduction in which the embryo of a female donor is put or transferred into the reproductive tract of a female recipient with the intention to increase the productivity as well as the desired traits of the animal or has the superior genetics. But why do we perform them? The application of embryo transfer in the reproduction of animals and even humans has several advantages based on several conducted research. As a matter of fact, this can be performed in any mammalian species. However, its widest application is done in cattle. One of the following advantages in performing the embryo transfer is it: first, it enhances reproductive potential of endangered species. It also enhances genetic diversity across a wide geographical region. Also, it enhances generation of offspring monotocous species. Next, circumvention of seasonal reproduction and assisted reproduction for infertility in humans. These are just a few of the advantages on why we perform embryo transfer. How do we know that it is successfully done? If you look at here in our illustration, as shown right here, a successful embryo transfer will involve the following. First, synchronization in the cycles of donors and recipients. Next, superovulation takes place. Then, followed by natural or artificial insemination of the donor. Then flushing, followed by the recovery of embryos from the donor and the maintenance of viable embryo in vitro. Lastly is the transfer of embryos to our recipient females. If all of the following are observed, then we can say that our embryo transfer is successfully done and performed. Before we move to our next topic, let's know what superovulation is. As shown in this picture here, when we say superovulation, this refers to the release of many oocytes during a single estrus period through a treatment of a female with gonadotropins. The application of superovulation is not only limited to, but is also used in humans. Here's the major steps of embryo transfer in mammals cow model. The goal in here is to make sure that our donor and recipient are in synchronization at the same stage of the estrus cycle. In this first step of superovulation, with a hormonal regimen that induces estrus, we then treat our recipients with it so that it will occur at the same time as the donor that has the superior genetics. In this second step of superovulation, we initiate the hyperstimulation follicular development by injecting gonadotropins to our donor. While the goal in here is to ensure that the best genetic combinations and fertilizations are generated as possible. In this third step of superovulation, we made use of very viable sperm and inseminators who had received extensive training and are experienced. While in this fourth step of superovulation, for the collection, we injected local anesthetic first, then inserted a catheter and introduced a flushing medium into the uterus of our animals. Further, in this last step of superovulation, with the use of a transfer pipette, we place the embryo into the uterine horn to achieve pregnancy for our recipient. Here's a snippet, an illustration of oocyte collection from ovarian follicles for in vitro fertilization. Also, we have here an illustration of how and why embryo viability must be maintained in vitro. As you can see here in our illustration, in vitro maturation of oocytes, the in vitro fertilization of oocytes, and the in vitro culture of oocytes from the ovum of our donor animal going to our recipient animal. But what is in vitro? The term in vitro refers to outside the living body and in an artificial environment. As you can see here in the images, the embryos are stored in an environment that maintains their viability.